Hello all and welcome back. So today we are going to discuss a practical topic from third year, and mostly in some institutes, even in masters, you have isolation of aflatoxin producing organism with extraction and detection of aflatoxin. Okay, so already a video, theoretical or say conceptual video just on aflatoxin is uploaded on my channel. Those are my handwritten notes you can go through. And yes, let's start with the practical topic. So first you need to isolate aflatoxin producing organism, that is actually a fungus or mold. Okay, so you will find this practical a little bit similar to isolation of molds. Okay, so yeah, the process is simple for isolation of fungi. So mostly aflatoxin is produced mainly by two molds, that is Aspergillus flavus and Aspergillus parasiticus. Okay, aflatoxin is a type of mycotoxin. Myco stands for fungi, so it is a fungal toxin or toxin produced by a fungi. Mainly these two molds, that is Aspergillus flavus and parasiticus, they are known for aflatoxin production, and they are widely uh, found on crops or grains or any decaying um, perishable food items. Okay, so of which Aspergillus flavus is very common and is widespread in nature and is most often found with certain grains and are grown under stressful conditions like drought. Okay, so we know that molds are ubiquitous, that is they are present everywhere and are easily found in soil, decaying vegetation, hay or grains, hay and grains. Hay is dried grass. Okay, and it and invades all types of organic substrates whenever and wherever the conditions are favorable. So what are the conditions? They actually need high temperature and high moisture content. Okay, so that is when they grow very easily and can spread throughout your crop. Now aflatoxin, there are at least 13 different types of aflatoxins which are known, but the major aflatoxin which are of concern are B1, B2, G1, G2. Now they are usually found together in food and feeds in various proportions. Okay, that means you may find combination of blue aflatoxin and green aflatoxin together. Okay, mostly they are found together in proportions, but B1 is most predominant and most toxic. When these aflatoxins are analyzed in by using a thin layer chromatography, they separate into individual components, and that is how you detect their presence. Now why they are termed as B and G? But because B aflatoxins, they fluoresce blue, and green aflatoxins, they fluoresce green under UV light. Okay, so after performing thin layer chromatography, you need to check the presence of aflatoxin under UV light. Okay, then aflatoxin M is another kind of toxin which is also very significant, and you may get a question in your Viva. Okay, so aflatoxin M is a major metabolic product of aflatoxin B1. Okay, it is a metabolic product of aflatoxin B1 in animals and is usually excreted in milk and urine of dairy cattle and other mammalian species that have consumed aflatoxin contaminated food or feed. Okay, now these are some structures which I think you may require to remember if in case you get a question in your Viva that can you draw the structure of aflatoxin. Okay, so there is very slight difference in case of the structure if you observe closely. Okay, so these are for B1, B2, G1, G2, and as we just read that aflatoxin M is another derivative of B1 aflatoxin. So these are the derivatives which are found in milk. Okay. Now another one is fate of aflatoxin M1 in raw milk. Okay, so Not just B1, B2, G1, G2 are important, even M1 aflatoxin is important because it is transferred to milk, okay, by the feed. If the feed is contaminated and the cattle eats that, then it carries or carryover contamination occurs when the moldy commodity has been eaten by the animal, and thus the toxin gets easily transferred to the milk. Now, when cows ingest contaminated feed, aflatoxin B1 is metabolized into aflatoxin M1 and M2 and is excreted in milk. Okay, so aflatoxin M1, it actually survives heat treatment. So pasteurization or sterilization of milk has very little impact on the reducing levels of this aflatoxin. Okay, the toxin is not very fat soluble, so it does not readily get transferred to your cream and butter, but some concentrations can be expected into other low fat products such as yogurt and soft cheese. Also, where milk is used as ingredient for other products like say infant formula, vigilance is again required concerning the potential aflatoxin M1 contamination. Now, what is vigilance here? So it is keeping careful watch on the possible danger of aflatoxin M1 contamination. Okay, because you are uh, using milk for infant formula, which are to be given to your babies. Okay, so that is also a very important standard that the company has to meet, that there is no presence of M1 aflatoxin. Okay, so some other mycotoxins can also be transferred from animal feed to milk, and there is some recent evidence of ochratoxin A, which is abbreviated as OTA. It is also been found in milk samples from different parts of the world. Now about aflatoxin B1, which is a very potent carcinogen in many species, including non-human primates, birds, fish, and rodents. Okay, so that is why we consider it as very toxic, okay, very dangerous aflatoxin. Now, studies have shown that this aflatoxin requires metabolic activation by exerting its carcinogenic for exerting its carcinogenic effect, and these effects can be modified by induction or inhibition of mixed function oxidase system. Okay, so to exert its carcinogenic effect, the aflatoxin has to be metabolically activated. Okay, the adverse effects of aflatoxins on humans are categorized in two forms. That is, first is acute aflatoxicosis, that is primary, and it results when moderate to high levels of aflatoxins are consumed, and episode of disease may include hemorrhage, acute liver damage, edema, alteration in digestion, possibly death also. Now, chronic aflatoxicosis, it results when low to moderate levels of aflatoxins are consumed, and the effects are generally subclinical and difficult to recognize, and some of the common symptoms are impaired food conversions and slower rates of growth. Okay, so these are the two types, or we can say forms of aflatoxicosis, and you can get a question in Viva on this as well. Now let's move towards isolation of aflatoxin producing organisms. Okay, so we are focusing on fungi. So you will need sterile Zapadox agar plate, okay, Lactophenol Cotton Blue for staining, and as a sample, you may collect some peanuts which show brownish green fungal growth. Okay, so you can use that as a sample. Now, if you're using peanuts as if they have very good amount of fungal growth on them, then you can use this peanut sample directly for inoculation. Okay, and if there is enough fungal growth, then only. If not, then you need to enrich these peanut samples. Now, how will you enrich? We need some good non-contaminated peanuts, okay, and then in a sterile flask under aseptic conditions, put some non-contaminated good peanuts with the contaminated peanuts with which has fungal growth. Okay, dampen a little by using a sterile distilled water, and you keep your flask in dark place, which is to be kept undisturbed for 3 to 4 days. Okay, so what will happen? Your fungal growth will now spread onto your good non-contaminated peanuts, and you will have good fungal growth, or even we can say a fungal mat can be observed upon your peanuts. Okay, now after that you need to incubate, you need to observe for isolation and growth. Okay, now proceed for identification by using lactophenol staining and colony morphology. So this is about your isolation of your fungi. Now about methods for preparation, or sorry, production of aflatoxin. So inoculum preparation. So once you have isolated your aspergillus culture, now you use that isolate, and it is to be inoculated on sterile Zepacox agar slant, and is to be incubated for 7 days. Now for why for 7 days? So that you will get a heavy crop of conidia, green conidia. We need the spores. Okay, so 3 ml of 0.005% of twin 80 detergent is added to the slant, so that you will it will help you to get a uniform spore suspension. That is why we add twin 80. Now the spores are scraped with loop, and the slant is shaken to give uniform suspension. This spore suspension was used to inoculate medium for production of aflatoxin. Now you need this spore suspension to be inoculated for production of aflatoxin. Now we have the isolate, we have the spores from the aspergillus. Now we are using that to produce aflatoxin. Now for that, solid state fermentation is used. Now media used is rice mill. So what you do, in 250 ml of flask, sterile flask, 25 ml of tap water is added to 50 grams of polished water, polished rice. Okay, then mixture was kept on shaker for 2 hours at 160 rpm at 30 degrees Celsius. Then flask was then autoclaved at 15 psi for 20 minutes. Okay, then 1 ml of spore suspension was used to inoculate this medium. Then flask was incubated at 30 degrees Celsius on shaker at 160 rpm for 7 days again. Okay, so what you did, you added some 50 grams of polished rice with 25 ml of water in a flask. It was kept on shaker so that your rice soaks some water, and you autoclave it. So now your rice meal is ready. Now you inoculate your spore suspension, and again you incubate for 7 days. Why? So that your fungi will grow properly. There will be a heavy growth of your fungi, and it will produce aflatoxin. Now care was taken to prevent the rice grains from adhering to each other by adding sterile water every 24 hours. Okay, so now what happens on shaker, due to continuous rotation, your rice grains, as they are slightly sticky, they can adhere to each other and can form a small ball. Okay, rice ball. So we don't want that. So what you do, you add sterile water every 24 hours. Okay, now greenish fungal growth was observed after incubation for 7 days. Okay, so you have produced your, we assume that your isolate has produced your aflatoxin. Another media or another method is broth fermentation, where media used is peanut mill. Now in again 250 ml of flask, 1 gram of peanut powder was added to 100 ml of Zepacox base, okay, or media. Now flask was then autoclaved at 15 psi for 20 minutes, and then 1 ml of spore suspension was inoculated. Again this flask was incubated at 30 degrees Celsius on shaker at 160 rpm. Okay, same as we did for solid state fermentation. Now here also your fungi will grow, and it will produce aflatoxin. Now about extraction. Now extraction from rice mill. So directly what you can do, you can observe the flask under UV torch. The wavelength is 365 nanometer, and growth and exudate on rice grain showed some blue fluorescence, confirming that culture um, that you have inoculated has produced aflatoxin, or it is a aflatoxin producer. Now 50 ml of sterile distilled water was added to the flask. Flask was again kept on shaker for 15 minutes, and then 100 ml of chloroform was added to the flask, and flask was again kept on shaker. The mixture was filtered through filter paper to remove the cell mass, or alternatively you can centrifuge, or directly centrifugation can be carried out. Okay, so a simple way is you can filter using filter paper, but you should be careful because it is a toxin. Okay, wear your hand gloves, mask, and work properly. Then filtrate which was obtained was transferred to separating funnel, um, mixed thoroughly and allowed to settle so as to separate the aqueous and chloroform layer. Okay, by using a separating funnel, the filtrate was transferred and allow it to settle so that your aqueous, that is water, and chloroform, it separates into layer. Now chloroform layer was passed through anhydrous sodium sulfate taken in funnel, and clarified filtrate was collected in evaporating dish and allowed to evaporate at 30 degrees Celsius, and then residue was obtained. Okay, so now this residue is what is your aflatoxin. Now extraction from peanut meal. Again here you can directly observe your flask with UV torch at 365 nanometers. Then broth showed blue fluorescence, confirming the culture to be a aflatoxin producer. Now broth is filtered to remove particulate media components and cell mass, or you can go for centrifugation. Now filtrate was treated with hexane. Okay, for rice meal we used chloroform, here we are using hexane to remove the contamination of other fats. Okay, as aflatoxin is insoluble in hexane, but there we used chloroform where it was soluble. So don't forget, first here you need to add hexane. Then mixture was transferred to separating funnel, mixed thoroughly and allowed to settle so as to separate the aqueous and hexane layer. Then the aqueous layer was collected and now you treat it with chloroform. Okay, as it is a broth meal, or we can say liquid state fermentation, what happens, there are some small components which are present, say from peanut meal or from your media, and they can interfere, so we need to treat our filtrate with hexane. Okay, so this is the difference between solid state and liquid state fermentation extraction procedure. Now once you have treated it with chloroform, mixture was again transferred to separating funnel, and mixed thoroughly and allowed to settle so as they separate in two layers, aqueous and chloroform. The chloroform layer was passed through anhydrous sodium sulfate taken in the funnel, and clarified filtrate was collected in evaporating dish and was allowed to evaporate at 30 degrees Celsius, and the residue was obtained. So this was about extraction. Now let's move towards detection. So a simple method that is used for detection is thin layer chromatography. Now for this you need a standard aflatoxin solutions for B1, B2, G1, G2. Then TLC apparatus, where TLC glass plate saturating chamber is required. Solvent system of chloroform is to acetone, that is 8.8 is to 1.2. Okay, you need to remember this ratio. Then UV torch. Okay, so why chloroform and acetone? Because our aflatoxins, they are soluble in chloroform, acetone, even methanol and benzene. Okay, remember that's the reason why we extracted our aflatoxin by using chloroform. Now the procedure. So residue obtained from both extraction procedures were suspended in 0.5 ml of chloroform. So you need to spot your sample, so that's why we need the extraction powder or the residue that we obtained, we need to suspend it in chloroform. This extracted sample was spotted on silica gel plate along with the standard. The silica gel plate was dipped in saturated solvent system jar, and after allowing the solvent to run the sufficient distance, the plate was removed and the solvent front was marked. Okay, immediately you have to do this. Then after air drying, the plate was observed under UV torch and the fluorescence was detected and compared with that of standard, and the distance traveled by the standard as well as your test sample is measured by keeping a scale aligned to plate, by taking care that you are not getting exposed your any body part to UV rays. Okay, by comparing the RF value of the sample with the standards, the type of aflatoxin that is produced in your sample is determined. Okay, so very simple procedure. So now here you need to remember the principle of TLC. Okay, so here is a simple diagram that you may need to draw in your practical exam. So you have the first TLC chamber, this is your silica gel plate, then this is the solvent front, these are the toxins which are separated. Okay, then ink spots, the spots where you have marked and inoculated your samples, and this is the solvent. Okay, so after running your TLC, you remove your plate, let it dry, and then observe under UV torch. Then you have to measure RF values and you need to report. So now this is a sample image where in standard you have all the four aflatoxins, that is B1, B2, G1, G2, which are separated properly and you can see those. In case of your test samples, you can see one, the first one has only B1 toxin and second one has both B1 and B2. So it depends on your culture and the proportion can vary. Okay, so you need to measure your RF values of standard and sample, and then you need to determine which type of aflatoxin was observed in your sample, or by which was produced by your isolate. Okay, so so now let's move towards Viva questions. So we have done isolation of aflatoxin producing organism, and even we have studied the extraction and detection of aflatoxin. So depending on all these three topics, these are some Viva questions that I could think of. And yes, the examiner can ask you anything depending on your answer, and he can link the another question, but still you should be prepared for at least these basic questions. Okay, so what is aflatoxin? This is the first and very basic question. So it is a fungal produced or toxin produced by fungi, or fungal toxin. Then what are the major types of aflatoxin? So B1, B2, G1, G2, and then comes the derivative, that is aflatoxin M1, M2. Then what is aflatoxin M1? So it is derivative of B1, and it is different as it is a metabolic product of B1. Okay, there is difference in the structure as well, and it is mostly found in milk or urine of the dairy cattle. Then which is the most toxic form? So it is B1. Then name two molds which are known for aflatoxin production. So that is Aspergillus flavus and Aspergillus parasiticus. Then B1 and B2, they fluoresce dash under UV light, so they fluoresce as blue color, and G1, G2 fluoresce green color. What are the two general forms of aflatoxicosis? So that is acute and chronic, and I have covered this in my video. Then aflatoxin is soluble in, so it is soluble in chloroform, methanol, acetone, and even benzene. Okay, then what is the role of hexane? So it is used to remove some interfering small components from the broth fermentation procedure. Then what is RF value? So it is the retardation value, or it is also known as retention value, and the formula is also discussed in the video. Then which solvent system was used? So we used chloroform is to acetone, that is 8.8 is to 1 is to 2, that's the ratio. And what was the wavelength of UV torch you used? So the wavelength was 365 nanometer. Okay, so this is all you should be knowing. Okay, the questions I have listed here, all the answers are covered in my video, so watch it again if you think you have missed out answer of some questions. Okay, So everything is covered here. So thank you for watching. I hope this video is very helpful to you all. You like my video, give a thumbs up to my video, do share my videos with your friends, and yes, don't forget to subscribe to my channel and hit the bell icon so that you will get notified on each new video that I upload. Okay, so I hope this practical series is helping you out, and be prepared for your Viva.